Hello, everyone. Thank you for joining us today. My name is Kathy Taylor, and I'm a chemist here in the Botanical Review Team in the Office of Pharmaceutical Quality, and I reside in the Center for Drug Evaluation and Research. And in today's webinar, we're going to talk about cannabis clinical research, and we're going to focus on drug master files and quality considerations. So let's first go over our learning objectives. The first one that I want to make sure you guys all gain is understanding of FDA's role in regulating cannabis products and the Farm Bill's impact on our FDA authorities. We'll go over some explanation of general botanical drug development and how that process works. I'll also elaborate on best practices for conducting clinical research with cannabis and cannabis-derived compounds. We're also going to learn the who, what, when, and how of drug master files, which we'll refer to as DMFs. Then we'll discuss how to use the DMF pathway in cannabis clinical research. We'll also present some examples on how to use those DMFs. And then I'll provide some FDA resources. A quick overview. We're going to go over FDA responsibilities and authorities just so everybody's on the same page. I know we might have some people on the line who've never interacted with FDA, and so at the beginning we're going to go over some really high-level FDA understanding. So for those on the line who have interacted with us, hang on because we're going to get into some more of the technical details later on, but it's really important to lay the groundwork at the beginning. We'll also talk about the 2018 Farm Bill's impact on FDA authorities and then FDA's role in the regulation of cannabis products. And then I'll talk about cannabis drug development, botanical raw materials, and then I'll actually delve into our new cannabis drug development guidance that's in draft, and then we're going to talk about those drug master files, and then we'll wrap it up all at the end. So for those who are new to FDA, our mission is to protect public health, and one of the ways that we do that is we ensure that the food we regulate is safe and wholesome and sanitary, make sure that all the labels that are on there are identified properly. We ensure that drugs for humans and animals, as well as vaccines and other biological products, medical devices intended for human use, are all safe and effective. We also ensure cosmetics and dietary supplements are safe and that they have proper labeling. And we advance public health by helping to speed product innovations. And lastly, we also regulate tobacco products. So you may have seen this slide in other presentations related to the FDA. It's a nice overview of all the different products that we regulate, and sometimes people don't realize how many product categories the FDA regulates. So these are FDA's responsibilities, but today we're mostly going to focus on the drugs in the FDA responsibilities. And I wanted to make sure everyone has a good understanding of FDA authority. So I want in your mind to picture your 7th or 8th grade biology class and that skeleton hanging in the corner of your biology class, and think of those skeleton bones as the Federal Food, Drug, and Cosmetics Act, the FD&C Act. Those bones are the federal laws that were enacted by Congress, and there are other federal laws along with that establish the legal framework with when FDA actually operates. Now, on top of those bones are the FDA regulations. Those are our muscles and our tendons. They help move our bones. Those are actually the regulations that are based on the laws that are set forth in the FD&C Act. And those can be found in Title 21 of the Code of Federal Regulations, 21 CFR, which we will reference a few times today. So in order to move those bones of the FD&C Act, we need the FDA regulations, which is 21 CFR. And then on top of that, we have the outer part of the body, which is our skin, which changes all the time with the seasons, with age, and that's what FDA guidances are. FDA guidance is something that changes over time, and it's essentially FDA's current thinking on a regulatory issue. And over time, as we learn more, as the science advances, we change and update those guidances. So one thing that occurred, I'm sure we've all heard about it, is the 2018 Farm Bill. And some people ask us, how did that impact the FDA authorities? Well, the good news is that under the Farm Bill, our authorities, which are in the FD&C Act, which we just talked about, and Section 351 of the Public Health Services Act, were specifically preserved by the Farm Bill. So that means cannabis, cannabis-derived products, including products containing CBD, are subject to the same authorities and requirements as FDA-regulated products containing any other substance that's non-cannabis. Some of the FDA authorities that we have include scientific and regulatory support for research on potential therapeutic uses of CBD products, approval of CBD drug products that are safe and effective, regulation of CBD products, and then enforcement actions as necessary against volatile CBD products, particularly those that present serious human or animal health risks. And I wanted to remind everyone that many products containing CBD are illegal under the Food, Drug, and Cosmetics Act. It is illegal to make a therapeutic claim unless an approved drug, and it's illegal to put it in food or dietary supplements. And our FDA jurisdiction over other products, remember those product categories I showed you over the map of the U.S., it's heavily dependent on the facts and the circumstances surrounding those different product categories. So an example is like lotions and soaps and other cosmetics containing CBD might be lawful depending on the circumstances. Same things go with CBD and vaping products. It could fall outside FDA's jurisdiction unless the vaping product makes a specific therapeutic claim. So it's very important that we get all of those facts and the circumstances around the wide variety of product categories that FDA has jurisdiction over. Now, one thing I wanted to bring up to those who are newer to FDA, and I know the veterans know all about this, but we had a public hearing about cannabis and cannabis-derived compounds on May 31st of 2019. It was very well attended. We had over 100 speakers with over 4,500 comments submitted to the initial docket. And if you happen to miss that, you can click that More Hearing Information link and learn all about it. And I wanted to remind everyone here today that we have reopened that docket indefinitely. And so we encourage you and we appreciate any comments, data, information related to CBD be submitted to that public docket. So you can click on that link at the bottom to learn more. Now, from that hearing, we had a lot of information provided to us, and it was important that our team continue to work together to help solve some of the challenges that our stakeholders are facing. And so here's a quick org chart of FDA. And as you can see, we have a huge team that across the agency works together on cannabis issues, and all of the centers essentially are working together to resolve these issues. And as a quick reminder, I'm on the Botanical Review Team, which is in the Office of Pharmaceutical Quality, and I live here in the Center for Drug and Evaluation Research. So all of our centers are working together, to resolve a lot of the challenges and try to find solutions that the stakeholders are talking about. Now, one thing that we did create after the public hearing is this new cannabis website. Down at the bottom, you'll see that there's a link that shows you where you can go, so you can click here anytime. And you can follow this link, and when you go to the website, this is the first visual that you will see. And here in the middle, we have cannabis, and then on the left, we have cannabis-derived compounds, which is where we're going to focus today. And then on the right, you have what we call cannabis-related compounds, which we'll briefly touch on, but the focus of today's conversation will be on our cannabis-derived compounds. So just as a reminder, the cannabis sativa L plant contains over 80 different naturally occurring compounds. Many of you have probably heard of cannabinoids. The two most well-known are cannabidiol, which is CBD, tetrahydrocannabinol, which is THC. And these cannabis plants are grown to produce varying concentrations of cannabinoids like THC and CBD. And these different plant variations are called cultivars. And all that means is humans have intervened to cultivate different varieties of cannabis plants, and they choose specific points of the plant that they want to either magnify or they want to lessen. And so first on the right-hand side, let's talk quickly about our cannabis-related compounds. How we categorize these in our current thinking is that these are synthetic compounds that are created in a laboratory. They can be used to manufacture drug products. Some are synthetic compounds, but some might also occur naturally in the plant. some might not. A few examples are the synthetically derived dronabinol, which happens to also be naturally occurring, and then nabilone, which is completely synthetic. And the agency actually approved 3 synthetic-related drug products, Marinol and Syndros, which are both dronabinol, and Sesamet, which is nabilone. Now, what we really want to focus on is this cannabis-derived compounds. So these are compounds occurring naturally in the plant, like CBD and THC, and they're extracted directly from the plant using traditional plant chemistry techniques, and they can be used, again, to manufacture drug products. So an example of this is obviously the CBD and THC that we've been talking about, and if you look in these red circles from the chemistry perspective, you can see that these compounds are very, very closely related to one another. And that's actually something to keep in mind when we're talking about how all of these compounds are connected to one another. An example of a highly purified CBD extract from the plant is actually the Epidiolex approved drug product, which is a highly purified cannabidiol drug product that is being used to treat rare forms of pediatric epilepsy and another seizure disorder that was just approved very recently. But I wanted to point out that in addition to CBD and THC, there are many other cannabis-derived compounds that you may not have heard of or you may not be as familiar with. A couple of the ones I wanted to point out are CBDA and THCA. Those are actually the acid forms of CBD and THC. They're what we would call the precursors. They're actually more abundant in the natural plant if you were to grow it outside, let's say, on a farm. The CBD and THC are actually formed from CBDA through a process called decarboxylation. And so beyond just CBDA and THCA, which are the precursors to CBD and THC, there are many other cannabinoids. A few popular ones are CBN, which is a degradant product often found, CBDV, CBC, CBG, CBGA, which is the precursor to CBG, and THCV. There's also a large class of compounds in cannabis known as terpenes. The 5 most common ones are listed here, and we'll talk about those in just a minute on our next couple of slides. And so here's the chemistry of a couple of popular cannabinoids, and I want to point out that in not just CBD and THC, but all of these compounds that are listed here, as well as the over 100 different cannabinoids that occur naturally in cannabis, there is very little known about these compounds. And so a big point of that public hearing that we had is we're encouraging and we would like to hear from you about any sort of scientific data that you have, any sort of preclinical trials that you might have, so you can help us close the gap on this huge scientific challenge that we're having, because people don't know enough about these compounds, and now people are wanting to use them in various investigational drug products, which we strongly encourage more investigational drug products using these different cannabinoids. So as you can see, we were talking about earlier that the structures are similar. So if you see here, CBDA and THCA have very similar looking structures. And CBN is not too far off because it's actually a degradant of CBD and THC. And the same thing here, you can see CBC and CBG and CBGA have similar type chemical structures as they're related to one another, as well as CBDV and THCV. So among these over 100 different cannabinoids, they are all have, some of them have connections to one another, while others may be a degradant product or maybe a precursor to one of the compounds you heard about. But we really want to encourage you to continue to study as many of these compounds as possible while using our investigational drug pathway. Some other compounds that are derived from cannabis are terpenes, and there are over 20,000 different terpenes that exist in nature. And so, interestingly, you may not have heard of these chemical names, but you have definitely smelled these compounds. So I'm going to start with limonene. The fresh citrus smell that you smell when you pull back the peel on an orange or a lemon or a lime, that's actually limonene. It happens to not just be in citrus, but also happens to be in cannabis. Pining is another one you're definitely familiar with. Think of a pine tree in your mind, the smell of pine needles. That smell is actually these aromatic compounds. And so there's over 100 different terpenes that naturally occur in cannabis. And these are the 5 most common: myrcene, limonene, linalool, caryophyllene, and pinene. But there are many others. And we also actually know even less about these in the clinical and the scientific literature. And so we do encourage you to continue to study these terpenes in addition to the cannabinoids that we were talking about previously, because we do have such a large scientific gap. And here at FDA, we want to learn all about the science, and anything that you can contribute would be extremely helpful. So let's talk about how you actually do cannabis drug development. So as I had said earlier, cannabis products that are intended for use under clinical trial with any claim of therapeutic benefit, with any disease claim at all, are in fact drugs. And so how would you submit that information to FDA? You would use what's called the Investigational New Drug Application, or IND. Or if you're not ready for an IND, you can request a pre-IND meeting with the specific clinical division of the therapeutic area of interest. So as an example, if you wanted to study an oncology indication, we actually have an oncology division. You would reach out to them and you would say, here is the specific type of cancer that I want to study. And they would direct you to the specific clinical team that would assist you in determining if they can grant you a meeting or perhaps give you some written response. This is a great tool to utilize for new researchers to FDA, or also veteran researchers to FDA who are starting some new research and would like to get specific feedback on their drug development plan prior to submitting the IND. As a reminder, once you do submit the IND, we have 30 days to review it. And so within that 30 days, we'll make a decision if something is allowed, called safe to proceed, or perhaps if there are some clinical hold issues. And so by having that pre-IND meeting, we can often assist you in answering a lot of the specific questions you might have. And there's no timeline on the pre-IND meeting. And so it does allow for some work that can be done on the research end. Now, beyond the IND application, you'll go through phases 1, 2, and 3 like any other drug product. Once you've reached phase 3, you can submit formally to the FDA and propose that we approve a new pharmaceutical that you've manufactured, and that is done via the New Drug Application product, the NDA. That's at the end. We're not going to focus so much on the NDA today. We're more going to talk about what happens in that IND phase 1, 2, and 3, because I think that's where a lot of us are living right now, and I think it's really useful that we get some more clarity on how to move forward in that space. So as a reminder, we treat products with cannabis-derived compounds as we do any other FDA-regulated product. Means same authorities, same requirements as any other FDA-regulated product that contains any other substance. So no special treatment. So let's dive a little bit into the IND. So when you're using a cannabis or cannabis-derived compound under clinical trial, you still must meet all of the FDA requirements for IND applications. This is a very long list, so if you click that link on IND applications, you can get all the specific details. But there are 3 broad areas that I wanted to bring up to you. The first one is the animal pharmacology and toxicology studies. That's your non-clinical and farm tox studies that you would submit. The second is manufacturing information. That's all the information on your botanical raw materials, your chemistry, manufacturing, and controls, which we call CMC. And the third is clinical protocols and investigator information. That's all the clinical study design. That's your inclusion, exclusion criteria. That's making sure that the appropriate medical staff is available for the design. So that is the types of information that would fall into those 3 broad categories. Now, it's important to know that as you transition through each phase of these clinical investigations, you must submit submission information to ensure the identity, quality, purity, potency, or strength of the investigational drug is maintained. And the amount of information that's appropriate to meet that expectation will in fact increase as you move from phase 1 to phase 2 to phase 3. So keep that in mind. And one resource that we have to help you is this Botanical Drug Development Guidance for Industry. It was recently updated in 2016, and that provides our current thinking on botanical drug development. It has lots of specific details and will help answer a lot of your questions. So if you've never seen it before, go ahead and click on that link when you have some time. And it focuses on quality controls of the botanical raw materials and their growing conditions. And we're going to talk about that now to make sure that everybody's on the same page. And so here we have our botanical raw material controls. We call it BRM controls. And up at the top, you'll see that we have our roadmap. So here's our roadmap. We're going to start at our growing conditions, going to harvest, transport, testing facilities, manufacturing facilities, and then to FDA. We're going to focus on these two right here, harvest and the GPS, or excuse me, harvest and the growing conditions. The growing conditions include what is called good agricultural and collection practices. These are established by the WHO. And we also will talk about harvest, which includes things like GPS location. So let's get into some details. One of the things that we're going to ask you when you come into FDA with your IND or even with your pre-IND is the plant species. So what varieties, what cultivars are you using? Then you'll also have to identify the botanical macroscopically and microscopically. You're going to have to tell us the GPS location of the farms or the indoor growing facilities, even if you personally are not growing the cannabis and maybe you're buying it from a vendor or you're buying it from a producer, you need to let us know where that cannabis is being grown. Cultivation and harvesting. Once you tell us where it's being grown, we need to know the actual growing conditions. So let's say it's an indoor facility. We need to know relative humidity, temperature, light, things of that nature. Stage of the plant growth at harvest. Are you growing the plant until it's 3 months old, or are you growing the plant until it's 10 inches tall? We need to know the difference of when you're going to harvest your plant and how many times per season are you harvesting your BRM and how many seasons do you expect in a single year. Obviously, this varies with where your cannabis or any other botanical is being grown in the world because seasons are varying based on the GPS location. We also need to know about processing. Once you harvest your BRM, how are you washing, drying, grinding that BRM? How are you controlling for foreign matter, your preservation procedures? How are you transporting it from the field or maybe a greenhouse somewhere? And then your storage, again, your preservation procedures once you have it at your storage facility. What is the temperature, relative humidity, light? What are those conditions? Do you have long-term storage? Do you have short-term storage? These are all things that you would need to provide to us so that we can do our assessment here in the Botanical Review Team where I reside, because we want to make sure that if you're creating a botanical drug product, that that raw material is as similar all the time, every time as you move it into your manufacturing processes, because when you're using live plant material, inherently there is variations. And so we want to make sure that these steps are maintained each and every time to reduce those variations as much as possible. So if we look back on our roadmap, whether you're using ginseng, turmeric, green tea leaf, or if you're using a cannabis leaf, this roadmap stays the same. The roadmap for all botanical raw materials is always the same. Cannabis is not treated any differently. And you can see all of these details, again, in that Botanical Drug Development Guidance for Industry. So you can check that out. Now, just in July, we published a draft guidance, which is titled Cannabis and Cannabis-Derived Compounds, Quality Considerations for Clinical Research. We've been getting a lot of feedback and asking for some more resources, and so this is something that we've provided to our stakeholders to help them gain some more insights. And so as I had mentioned previously, if you want to study cannabis or cannabis-derived compounds in a clinical setting, they should be under an investigational new drug application if the use is for humans. And so in this guidance, which is specifically only for human drugs, we don't talk about any other FDA categories, there are 3 main topics, and we're going to talk about them today. The first one is sources of cannabis. The second is resources for information on quality considerations. And then the third is the % delta-9 THC calculation. So the first one, remember we had just talked about botanical raw materials in our BRM roadmap. It's important that you must meet all FDA requirements to conduct human clinical trials, regardless of the source of cannabis or any other botanical product under study in the trial. That's part of the 21 CFR Part 312. As a reminder, those are those muscles and tendons in our skeletal model that we talked about way at the beginning. Now, something for those of you who are veterans, you know all about the National Institute on Drug Abuse, which we call NIDA, and their drug supply program. But for those who are new, the NIDA DSP has provided for many, many years the only domestic federally legal source of cannabis for clinical research. It is grown under contract by the University of Mississippi at the National Center for Natural Products Research. Now, when we had the 2018 Farm Bill, which you can click on that and it will take you directly to the law, there were some changes that occurred. And those changes now allow hemp to serve as a source of cannabis and cannabis-derived compounds for drug development if they do not contain delta-9 THC at more than 0.3% by dry weight. That is the limit that was established by the 2018 Farm Bill. You cannot exceed the 0.3% by dry weight THC limit. The 2018 Farm Bill now gives sponsors and investigators who are looking to use cannabis and cannabis-derived compounds in clinical studies new options that don't involve the NIDA drug supply program. So here we talk about kind of the 3 different places that we wanted to clarify for you since there's been some changes to the 2018 Farm Bill. So we just talked about how the NIDA drug supply is the only domestic federally legal source of cannabis that's over that 0.3% delta-9 THC limit for clinical research. The second category is cannabis that is at or under that 0.3% delta-9 THC limit may be used for clinical research. And the third category is anyone who is thinking that they may be using a controlled substance, or even if you're unsure, we encourage you to contact DEA and they can answer all of your questions regarding controlled substances and the Controlled Substances Act to make sure that you are in compliance at all of your facilities. Now, resources for information on quality considerations. I think this one is really useful because in all the INDs we talked about that you're expected to show that you can consistently manufacture a quality product. And we had talked about how you have to submit sufficient information to ensure the identity, quality, purity, potency, or strength of your investigational drug product. And you need to provide quantitative information, so something like a certificate of analysis, which I'm sure many of you are very familiar with. Something that you would include in that C of A would be data on phytochemicals such as cannabinoids, terpenes, flavonoids, any other compounds that you're actually seeing in your cannabis or your cannabis-derived compounds. So you could include those in that C of A that you would provide to the FDA. And you can refer to FDA and other ICH guidelines and guidance to help assist you when you determine your analytical method validation. And we do have some guidance documents on pharmaceutical quality that are available. You can click them after this and you'll be able to find some specific information on many topics involved in pharmaceutical quality generally and not just cannabis. And there are a few principles and documents that I wanted to point out to you. We're not going to talk about all of these, but the ones that are in the darker blue squares, I wanted to quickly highlight for everybody here today. One thing that I think is important and sometimes can be a challenge for researchers and investigators when they come in here is making sure that they have adequate characterization using the chemical fingerprint. There's lots of different methods that you can use for this, but making sure you have adequate characterization with chemical fingerprint is definitely a challenge that we have seen, and we wanted to point it out to you that there are resources available that can help make sure you get adequate characterization. Another, up in the right corner, You'll see we reference the USP Chapter 61 and 62. This is the microbiological information for non-sterile products. Specifically, if you're going to be using cannabis or cannabis-derived compounds in a clinical setting, it's important to make sure that you check out these micro testing and the criteria that USP has established, because it's very useful. And it is dependent on the route of administration, so make sure you look carefully at that. Another thing in the middle here, the two dark blue squares, is drug scheduling. If you do think that you might be using a product, again, that possibly is a controlled substance, or you're not really sure, it's important, again, to contact our colleagues at DEA. And keep in mind that if you go through the stages of IND, phase 1, 2, and 3, and NDA, there is a potential that you might have to do abuse potential assessment study, and that will be dependent on the compound or compounds you are using and if they have a potential for abuse. We have a staff here in CDER called the Controlled Substance Staff, and they would actually do that assessment, and they would let you know during the IND phase if they felt that that study needed to be done, which would be done on the researcher side. The next one is, we don't recommend pursuing approval of an NDA relying solely on published literature. And the reason we don't recommend it is because the published literature is missing, and there's a lot of scientific information specifically about products. And so if you were to rely solely on published literature, there will be a lot of gaps. As a reminder, you are required to provide, strength, identity, purity of your investigational product, and a lot of times that information is not in the published literature. So it doesn't mean you can't use it. We just don't recommend that you solely rely on it. So you're going to have to produce some of your own data as well. In the bottom left corner, I wanted to remind folks who are planning to use cannabis or cannabis-derived compounds in combination with a device, that product would be considered here at the FDA what we call a combination product, and it must comply with the CGMPs that are related to combination products, which can be a little bit more elaborate if you were not using a device. So wanted to bring that to your attention. And the last one on the bottom right corner is about impurity profiles. And so if you plan to use cannabis or cannabis-derived compounds or an extract that are coming from a botanical plant, that will have one impurity profile. Or if you're planning to use a cannabis-derived compound that is actually synthetically being derived, maybe more of that cannabis-related compound that we talked about earlier, then that will have a separate impurity profile. So those may be the same compound, but their impurity profiles will be different. And those impurity profiles will be evaluated during the IND stage, specifically by our CMC colleagues, and so they can provide insights. So the others are also equally important. I just wanted to point out these few to you today. Now let's talk more about this % delta 9 THC calculation. It's important to remember that any growing and manufacturing of cannabis that's used for research, it has to comply with the Controlled Substances Act and any DEA requirements if, again, you're over that 0.3% delta 9 THC by dry weight limit, because that's what was established by the 2018 Farm Bill. Again, go ahead and consult DEA if you think you might be using some controlled substances. Here at FDA, we think it would be useful to you as a researcher or investigator to calculate the level of delta 9 THC in any of your proposed investigational cannabis drugs. And we suggest that you do it early in development to gain some insights on potential control status. And again, as I mentioned, our controlled substance staff can help assist in that area, and we'll have some contact information for them at the end, so you don't have to worry about that. We also suggest that you work with reliable labs for all of your analytical testing during your IND development. And we might request additional CMC info during your drug development. And so keep in mind that as you move through those phases, we're going to be asking for more and more information. So it's important to establish a relationship with a reliable analytical laboratory at the beginning of your research. And this calculation for tetrahydrocannabinols may help you figure out the composition of the proposed raw material that you're using, the intermediate that either you are creating or maybe a third party is creating for you, the drug substance or the drug product, because those are all different phases of the manufacturing. So it's important to make sure that you're doing your calculations early on in your development so you can gain insights. And then it's important also to note that that 0.3% delta-9 THC dry weight limit is not appropriate from FDA's perspective as a limit for tetrahydrocannabinols as impurities in terms of quality control purposes and in your application submission, specifically for the CMC section. So keep in mind that that limit is set by the Farm Bill, but FDA currently, based on the information we have, we don't feel that it's actually an appropriate limit as an impurity. And those quality-related calculations that we've been talking a bit more about, those will be evaluated during the IND review process, and they will be specific for your IND. So no IND will be the same, and we will provide specific feedback to you. I'm sure everyone's aware of the USC interim final rule, so make sure you're referencing that when you're learning about your sampling and your testing methods for that delta-9 THC. They've established all of the methodologies. Providing that quantitative data we had talked about earlier, certificates of analysis. On your certificate of analysis, it's extremely useful to indicate that delta-9 THC value, whatever it might be, by dry weight in your botanical raw material. And the reason that we ask you to do that is because if you're taking that raw material, once it enters manufacturing, that delta-9 THC value will change up or down. It will be dependent on manufacturing processes. So please indicate it for the raw material, and then you can also indicate it in your drug substance or your final drug product. And that information will definitely be requested by our CMC colleagues. Again, make sure that you're providing detailed testing methods. Phase 1, we don't need as much, but phase 2 and 3, it is extremely important that you're providing those detailed testing methodologies to us and making sure that they're validated as you move forward. And when you're doing your calculations, one thing to consider is the USP General Notice and Requirements, the section on rounding rules. Make sure that you're calculating them according to those rules, because that's what we're using when we're doing our calculations. So just a helpful hint. And in general, not just for cannabis, but for anything, when you're doing the composition of a botanical raw material, the generic calculation is you take the amount of compound or compounds of interest that are present naturally in that raw material, and then you divide it by the dry weight of the botanical raw material prior to you using it in an extraction or any sort of manufacturing. So that's the general calculation that you could use, and you'll have to make it specific for your raw material, but that's generally a good practice to start with. Now, when you have your raw material, the next thing would be intermediates, and then beyond that would be finished products. And so what we recommend is that you calculate that delta-9 THC based on the composition of the formulation with the amount of water completely removed. That means you need to also remove any water that is found in any of your excipients in your final product formulation. Excuse me. This calculation should not be used for other purposes such as CMC. And here again, this is what the guidance looks like. And rather than me retyping it, I just wanted to show you that this is a specific example directly from the guidance. So for a solution-based material, such as an intermediate or an in-process material, these are the steps that we would actually recommend for you to do your calculation. Very quickly, we asked you to determine the density of the liquid formulation, convert it to a milliliter of formulation to mass units. You calculate your water content. You sum up the water content for all of those components. Make sure you're subtracting that from your total mass from step 1. And then you're going to calculate the mass of the delta-9 THC present in 1 milliliter of the liquid formulation. And then from that, you're going to calculate your percent delta 9 THC by dividing the mass of the delta 9 THC from that step 4 by the total water adjusted mass in step 3, and then you multiply it by 100. And so, as I had mentioned, there was that one specific recommendation for the solution-based material, but we also have some for solid oral dosage forms, tablets and capsules, which you can check out. And then if you're creating intermediates or byproducts during your manufacturing process that might exceed that delta 9 THC dry weight 0.3% threshold, even if you're starting with a material that meets that definition of hemp from the Farm Bill, those intermediates and those byproducts might be considered Schedule I controlled substances. And so we strongly encourage you, contact DEA, get some recommendations if you anticipate you might generate some of those intermediates or byproducts to make sure that you are in compliance. And lastly, once you have had some time to look over this drug development guidance, it is a draft guidance, which means we appreciate any comments that you might have and we welcome them. You can click on that public docket link right up here at the top, and it will take you to the place where you can submit comments. Some possible comments that you might have would include potential challenges with anything that we're suggesting, or topics you found useful or that were not useful, suggestions for improvement, any additional topics for consideration, and also if you have any sort of data or information that are related to the guidance that you'd like to share with us, we appreciate you providing those comments to the docket. And we do encourage you to share that draft guidance with your staff and your colleagues, your networks, for anyone who operates in the cannabis space so that they're aware of the information. Now, another thing that we learned when we had our public hearing back in May of 2019 is a lot of stakeholders said to us that they had some concerns about protecting their proprietary information. And they wanted to share information with FDA, but they had concerns about sharing proprietary info. And so we took this back and we talked internally. And we would like to have stakeholders consider utilizing our well-established drug master file pathway, which is called DMF. And I'm going to present that to you today so you can gain some understanding about utilizing this potential pathway. So what are drug master files? Well, DMFs are submissions to FDA that we use for you to provide us with confidential and detailed information about either your facilities, your processes, or articles that are used in your manufacturing, processing, packaging, and storing of human drug products. In this pathway, the information is shared only between FDA and the DMF holder, who we're going to talk about in just a moment. And DMFs, there's actually 4 different types, but today we're going to focus on the most common type, which is called type 2, and that includes drug substance intermediate, material used in the preparation, or the drug product. Now, a few highlights of what the DMF does: allows you to share sensitive data confidentially with FDA, allows parties to reference your DMF without you having to disclose the contents to those different parties. It's an electronic submission platform. It can be updated at any time. A DMF is neither approved nor disapproved. And there's no cost when it's submitted to support an IND or an NDA. However, in compliance with our GDUFA regulations, there is a fee when you use it submitted to support an ANDA or a generic drug application, which we're not talking about generics today, but it's important to know that there is that one distinction. Who can submit and use DMF? Well, anyone can submit a DMF who wants to communicate information to FDA about their facilities, processes, or articles that are used in the manufacturing, processing, packaging, and storing of the human drug products. So any of those blue, green, orange, and purple figures that are up there in the left corner, they would be the entities who are submitting the DMF, and they are called the DMF holder. Now, the FDA will only discuss the contents of the DMF with the DMF holder. So the DMF is our little red dot in the corner. FDA only talks with the DMF holder about what's inside the little red dot. Now, who can use a DMF? Anyone who's conducting clinical research under an IND or an NDA can use information in a DMF with permission from the DMF holder. Now, the DMF holder provides permission for FDA to conduct a technical review of what's inside that DMF via a letter of authorization called an LOA. So let's talk a little bit more about how that process really works. So if you wanted to utilize the DMF process, what you would do is you would first obtain a pre-assigned number before you submit the DMF electronically to FDA. Then, once you have your pre-assigned number, you collect all of your information, you submit the DMF, and it undergoes administrative review. That's just we're checking the boxes to make sure you've submitted all the information we've asked you to submit. Now, if your DMF is accepted administratively, we confirm that acceptance with you, who would be the DMF holder. And then we wait. We wait until a letter of authorization comes in associated with an IND or an NDA in this particular example. And then once we have that LOA to the DMF to support your IND or NDA, we then initiate what's called a technical review. So then FDA reviewers, like myself, my CMC colleagues, we will review the technical contents inside of your DMF to support a clinical trial, which is an IND, or an application, which is for an NDA. And if we then, once we've completed our review, we will then find your DMF either adequate or inadequate in support of the clinical trial under IND or the application under the NDA. Now, you can click here and learn all about the specifics of these detailed processes, but this is the general overview of how the DMF process works. Now, when are the DMFs actually reviewed? Again, we perform that cursory administrative review right at the beginning upon submission, making sure you have checked all the boxes. And then when that letter of authorization for the DMF is submitted to support an IND or an NDA, that will trigger our technical internal review. And it will only be the review of the DMF contents for that specific application. And the DMF is deemed adequate or inadequate. And we will alert the DMF holder of the adequate or inadequate status. Now, as a reminder, a DMF is never approved or disapproved. It's only adequate or inadequate. Now, as a DMF holder, you would be required to submit annual reports, and if there's any updates or changes to the technical content, you do have to submit that information regularly to FDA. So you do have to keep your DMF updated. Now, once that LOA is provided by the DMF holder to the researcher who wishes to use the contents in the DMF to help support that proposed clinical research, then we can do our technical review. But that letter of authorization is required. If an investigator does not have the LOA and they just write the DMF number in their IND, we cannot do a technical review to support that IND. So it's very, very important that that LOA is provided by the IND sponsor upon submission. And so we have the LOA. We've done our review of the drug master file. What happens if the DMF is considered inadequate to support the clinical trial? Well, the FDA reaches out to the DMF holder, and we will have a list of deficiencies. And the request will go to the DMF holder only to provide information to address those deficiencies. We will also inform the IND sponsor or researcher who referenced the DMF, and we'll say, this DMF is inadequate to support your clinical trial, and the researcher would need to speak directly to the DMF holder. We will never share DMF contents with anyone but the DMF holder. And so very quickly, this is the information sharing that's going on. And so DMF contents, again, is only shared between FDA and the DMF holder, so it only goes this way. And if they have deficiencies, we will reach out to DMF holder, say, hey, please provide XYZ information. DMF holder then provides that to the FDA. We will then do a review of that information and decide if it's sufficient to address the inadequacies and the deficiencies. FDA will inform the IND or the NDA sponsor researcher if the DMF they reference is inadequate, but we're never going to communicate contents of the DMF to this entity. Now, if the DMF holder wants to share something with the IND sponsor or the NDA researcher, that is at their discretion. FDA will never do that. So it's very important to note that this is the confidential proprietary information discussion only happens between DMF holder and the FDA. Now, let's go over just 2 quick examples so you guys understand how DMFs work. So we have Bob's CBD Emporium. Bob makes an extract of CBD from industrial hemp using a proprietary method that he and his team have developed. Bob's been contacted by 2 different researchers, Dr. Kathy Smith up at the top and Dr. Tanya Rhodes down at the bottom, and they would like to use the CBD extract in 2 different clinical trials. So Bob doesn't want to give them all of his confidential information because these 2 researchers would have to submit that information to FDA, because as a reminder, I would need to know all the information from our botanical roadmap, and my CMC colleagues would need to know all of the manufacturing information. So Bob has decided he's going to submit a DMF to FDA so that all his confidential processing, manufacturing info doesn't have to be given out to researchers who want to use a CBD extract. It's a great option for Bob. So Bob submits his DMF electronically, goes under that administrative review, and then we wait until a letter of authorization is provided to support a clinical trial or an application before we initiate that technical review. So Bob gets notified his DMF has been administratively accepted, and he receives acceptance for his DMF 123456. That's his number. Now, Dr. Kathy Smith, she wants to use Bob's extract for her clinical trial to study how it might affect participants suffering from anxiety. So Bob and Dr. Smith agree. He will provide the extract to her for her clinical trial. Bob provides Dr. Smith with the letter of authorization to his DMF. And then Dr. Smith writes up her IND application, and when she submits it to FDA, she includes the letter of authorization for DMF 123456 from Bob, giving FDA permission to conduct that technical review of the DMF contents to support Dr. Smith's research. Now, at the same time, Dr. Rhodes also wants to use Bob's CBD Emporium CBD extract. She wants to use it in a separate clinical trial. She wants to study how CBD might affect children with autism. She contacts Bob separately. They agree that he will provide her with the extract. And then Bob sends Dr. Rhodes an LOA to his DMF, which has already been accepted by the FDA administratively. And then Dr. Rhodes creates her IND submission, includes that LOA to Bob's DMF. That gives FDA permission to conduct a technical review of the DMF and its contents to support Dr. Rhodes' research. So let's talk about the benefits of using DMF as we wrap up here. The proprietary and confidential info, specifically the CMC info, the botanical raw material info, is protected. DMF holder doesn't have to disclose that confidential info to any of the researchers like Dr. Smith and Dr. Rhodes or any other collaborators if that DMF is provided to FDA. And then FDA never shares the contents with anyone. It's only between the DMF holder and the other entity. Excuse me, the DMF holder and FDA are the only 2 entities that will discuss the DMF contents. A DMF can be used by many researchers at one time. An LOA is required by each researcher to reference the contents, but you may reference more than one excuse me, IND under the same DMF. And there's no cost if you submit a DMF to support an IND or an NDA, but if you were to do it for the generic side, there is a cost associated. And then lastly, the DMF provides researchers with more options to conduct clinical trials using human drug products with cannabis and cannabis-derived compounds. So please check out some of our resources here. We have a draft drug master file guidance for industry. It goes into wonderful detail about all the different steps. We have a DMF webpage, which provides additional information about the DMFs. I think it's extremely useful for you to take some time and click through all of these web links. There's a list of DMFs that FDA updates quarterly, so as a sponsor or researcher, you actually go there and pick out a DMF holder that you would maybe want to collaborate with, and you can reach out to them. There's also different types of DMFs. We talked only about type 2 today, but you can check out the other types as well. If you are considering using this pathway, there are already submission resources. There are different templates that you can use. There's all this other information that's already collated for you on the FDA website, so I strongly encourage you to go there first. And if you have any questions, there's actually a mailbox that you can email to, and they can answer your questions and help you move forward using this pathway. And a few additional resources I wanted to show very briefly is this web page. If you haven't seen it, is our cannabis and cannabis-derived products, including CBD web page. It has 4 main categories: our consumer information, and then we have a lot of FDA communications, as well as our regulatory resources. Excuse me, this jumped forward. But the main point is our question and answer section. And we have about, I think, 26 or 27 questions on there, but I know it's being updated very regularly. So a lot of the frequently asked Q&As are there. So please go there if you haven't checked out this website. And if something is missing, you can send us a note and let us know. And if we get a lot of requests for a specific Q&A, we can go ahead and update this website. So make sure you check that out when you have some time. This is a contact for my team, the Botanical Review team. If you have any general inquiries, you can reach out to us. We also have a web page that's publicly available. So if you want to learn more about botanical drugs, you can always click here. And then we also have the controlled substance staff, which I had talked about earlier. They're the ones who are help liaise between FDA and DEA. So if you want to learn more about them and how to work with controlled substances, you can go to their web page. And then this is a very long list of all the FDA cannabis resources I just wanted to point out. We have the guidance we talked about, that public docket, an FDA in brief that went out with that guidance. We talked about that web page I just referenced, and then other things that we have worked on since the May 31st, 2019 public hearing. Our commissioner, Dr. Hahn, put out a statement in March, which is talking about how we work together with all of our stakeholders. And then lastly, that botanical drug development guidance, which we talked about on that botanical raw material roadmap. And lastly, we have our CDER Small Business and Industry Assistance, SBIA. They were gracious enough to help put on this webinar for us today. And so you can always get more information on their webpage and follow them on LinkedIn. And so we're very grateful that they have these webinars and learning activities available to all of our stakeholders. And as we wrap up here, I wanted to quickly summarize what we talked about. As a reminder, FDA authorities were specifically preserved by the 2018 Farm Bill, so that means nothing changes. Cannabis, cannabis-derived compounds are regulated just as any other FDA-regulated drug product is. We support and encourage strong science-based research for all botanical human drugs, including those with cannabis and cannabis-derived compounds. We have our quality considerations for cannabis and cannabis-derived compounds in our human drugs. That's in that draft guidance. Comments on the public docket, we very much appreciate those. You as a stakeholder can protect your proprietary information by using the DMF. Remember that the DMF can be referenced by more than one researcher at one time, and your content is always protected. It's just between you, the DMF holder, and the FDA. And if you submit the DMF in support of an IND or NDA, there is no cost. There's just that fee associated for generics. And then DMFs also might provide researchers with more options to conduct those clinical trials using human drug products with cannabis and cannabis-derived compounds. And we have all this information here, but if for some reason you can't find what you're looking for, please contact our CDER SBIA staff. Here's all their contact information. They are wonderful to work with, so please don't hesitate to reach out to them. And I do want to thank them again for this opportunity for me to share all of this information with you. So thank you very much, and I hope you have a wonderful day. Thank you for listening.